All right, I think we are going to go ahead and get started even as some more people are joining us in the room, but settle in with your coffee or caffeinated beverage of choice as we've got a packed agenda today. Hello, my name is Hannah Rosentrader and I'm a manager of special projects here at the National Alzheimer's Coordinating Center, lovingly known as NACC. Welcome to our virtual 2021 Fall ADRC meeting. We've got our clinical task force session up first, happening from 7 a.m. to 10 a.m. Pacific time. So translate that to the time zone of your choice. Without further ado, though, I would love to introduce Dr. Allan Levey, PhD and director of the Emory ADRC, as well as leader of the clinical task force. So everyone give a round of applause as we welcome Allan to the stage.
Thanks so much, Hannah, for the introduction. If you could let me share a screen, I'll just show a few slides. Welcome, everybody, particularly, and thanks to all of you, especially on the West Coast, for waking up early to join us. Hopefully you have your first cup of coffee in hand, and if you're on the East Coast, you're on your second. Let me go to screen share, and Hannah, make sure you can see my screen. So I'm just going to give a few introductory comments because, as Hannah mentioned, we have a very packed session this morning from the Clinical Task Force as we give you our updates. You'll be hearing from each of our 3 subcommittees and work groups: the Clinical Measures and Diagnosis Subcommittee, our Neuropsychology/Cognitive work group, and our Technology Subcommittee. First of all, let me thank the folks that you're going to hear from today. This has been an amazing team across all the work groups. The number of person hours that this group has been putting in on all of our behalf is really extraordinary and higher than I can count. Particular thanks to Greg and Cindy for leading the Clinical Measures and Diagnosis Working Group, Andy and Lisa in the Cognitive Working Group, and Rhoda in the Technology Work Group for helping organize the sessions today. Also want to acknowledge, since we last showed you the membership last spring, I want to thank Constance Laketsos from Hopkins, who's joined us in recognition of our need for additional psychiatry expertise as we try to embrace a better capturing of behavioral symptoms. And I also want to give particular thanks to the entire NACC team, who has really worked tirelessly across all of the work groups. At least 1 or 2 meetings a week on average have been going on since the last meetings, as well as Nina, Teresa, and Grayson from the NIA, who also participate in almost every single meeting. So thank you all for helping make this task force happen. Just as a reminder, a framework that has been useful for the Clinical Task Force Committee is what we finally refer to as the Greg Jicha atomic model of the UDS. And the nucleus is really what we're working on in the atomic model. So the nucleus is the nut, the core of what we all do across the Alzheimer's centers. That is really critical, as we all know, to enable harmonization. But it's also a good reminder that this becomes a critical platform for us because it is a platform not only for our site-specific research, which is what motivates so many of us, but also the team science that can happen and the increasing consortia that rely on the uniform data set, including Diane, Mark, VCID, LEADS, the DLB Consortium, all FDD, and not to forget major studies like ADNI and our genetics initiatives, et cetera. The power of what we do as a consortium of Alzheimer's centers is only going to grow in the future as data becomes more and more important to, I think, drive the field forward. And we've also been cognizant as a task force that our UDS-4 has to do a better job of understanding the heterogeneity of disease, and it needs to be able to address the needs of diverse populations and support all these studies while allowing harmonization. So just keep that in mind as you hear about our UDS-4 updates today. There are also, I just want to remind folks of the guiding principles that our task force has been using. First and foremost, our core battery, we all recognize has to enable the best science. We know we've been charged, and we all agree that the core battery needs to be streamlined where possible to reduce the burden both on our participants as well as on our staff. On a critical measure, as I just mentioned, is harmonizing the core measures and ensuring that we have sort of systematic, consistent ways that forms are being completed. And then I think one thing that's really critical for all of us to keep in mind as we go forward, unlike past versions of the UDS where there have been lockdowns where it never changes, we recognize that UDS 4.0 will quickly become UDS 4.1, 0.2, et cetera, because the field is changing very rapidly. And we as a group of Alzheimer's centers need to be dynamic and adapt, just as we've shown our ability to do so over the last year or two, with all the challenges coming at us. And we know that there's a rich data set that we need to preserve where possible and ensure opportunities for harmonized new data with the older. So we certainly will continue to prioritize site-specific measures, so there's time for that to do that. Increasingly, we'll have groups of centers collaborating clearly, so we don't need to collect everything in detail across all centers. And we want to make this process easier. So I think in future iterations, you'll also see more and more technology to help in delivery of the collection processes in a more efficient manner. So just a word on what we're going to talk about this morning from our 3 work groups. First, you're going to be hearing from the Clinical Measures and Diagnosis Subcommittee. We've reordered the presentations a little bit. Greg, thank you for doing an amazing job striving for consensus across a large group of centers. Consensus is never easy, but you've been masterful at doing what is possible. So one of our curveballs has been obviously the introduction of Aducanumab and how that's going to impact the Alzheimer's disease research centers. So first, Greg's going to provide the consensus statement process review and update. And then you're going to hear from, I think, Suzanne, Teresa, and Jeff. I think Suzanne may be leading that discussion. With the recognition of Aducanumab and more drugs to come, and their impact on biomarkers and potentially disease modification, we all recognize that we need to better account for which of our participants are on study medications. And so this is a new concept for the UDS, how to capture that information. You'll hear about that. And then the real meat of the morning from the clinical measures group is going to be from Cindy and Greg. So we're really thrilled to bring you all our recommendations for UDS version 4.0. And we want to make sure we have plenty of time for those guys to walk us through that. The Neuropsychology Subcommittee presentation will be brief. We all know they've done their heavy lifting with the pivot to COVID and remote assessments. Work certainly is continuing to progress, and Lisa and Andy will give an update. And then we also have a very exciting presentation from the Technology Subcommittee. And I want to thank our speakers in advance, Kate, David, Vijaha, and Giannis, who are highlights of a lot of work that's been going on in the subcommittee. We all recognize that digital technologies and technological advances of other types are just going to be increasingly important in the future. They're really going to enable a whole new form of data capture. They and other work ongoing across all the centers will continue to lead to a proliferation of data which can be mined, and we have to make sure our Alzheimer's centers are prepared, and more importantly, that we're leading the way. It's so much of the future for care and research for Alzheimer's disease and related disorders, and this technology subcommittee and the presentations you will hear, I think, are going to start positioning us well for that. And so with no further ado, let me turn this over to Greg, and thank you, Greg, and I'll stop screen sharing.
Fantastic. Thank you, Andy or Allan, for that summary of where we are in the grand scheme of things. And yes, it has been a lot of work, but always, and I think I speak for everyone, a pleasure to be able to contribute in the way that we have. And one of the major contributions, which has really come about over a very short period of time, really all of us being thrust into the reality that Ajikanamab has been conditionally approved by the FDA. There was a call for centers to utilize our expertise in the field, and pulling together all centers is not always easy, but this is the Aducanumab Appropriate Use Working Group. And as you can see, every center is represented, and I'd like to thank all of the members. We really have accomplished a tremendous amount in a very limited amount of time. Welcome Duke and South Texas ADRC. We're sorry that you missed out on these early discussions, but we're certain to include you in all future work groups. And also a special thanks to the other chairs of the CTF subcommittees, and our NIA colleagues. On the basis of disclosure, I will comment that Nina, Teresa, and Grayson, while supportive of the CTF, did not play any role in content or discussion within the Ajikanamab working group. This is truly representative of the expertise at each of the centers. So what is that expertise? You know, I was really amazed to look at this, taking the members together, 661 combined years of ADRD research and clinical experience on a timeline that would take us back to the bubonic plague. 497 combined years of ADRD clinical trial experience takes us back to Giovanni de Verrazzano, who discovered the New York Harbor in that year. Anti-amyloid experience takes us back to the first ever State of the Union address by George Washington, and 72 combined years of Ajikanamab experience takes us back to a year notable for the creation of NATO, introduction of the Polaroid camera, the VW Beetle, and the MBA, importantly. So a wealth of experience. So we may all ask, and I know, you know, we're always interested, what are the other centers doing? I know where my center is. And this is a summary from just a little over a week ago on where the ADCs are in the grand scheme of things. As of yet, it doesn't look like anybody has this up and running, but most centers are planning on moving this forward. And like my center, it may be going through formulary approval committees. The debate is still out on coverage for it. And so there's a lot of uncertainty in the field. Sites, though, gearing up and looking at this, you know, have expressed that prescribers will likely be restricted to experts like ourselves. And one of the concerns that came up through the group, and we'll talk about this a little bit later, is safety, and whether or not we have that backup support. Do we have the radiologists clinically that can recognize ARIA in whatever form? And I was shocked to find that almost 20% of the centers were fairly confident that they did not yet have the radiologic experience to roll this out in a safe clinical fashion. So we'll start with what we didn't do. And we did not debate Ajikanamab approval. We all know this history, and I'm not going to go through it. It was not about that. It was about understanding the reality that Ajikanamab is here, that many patients and caregivers are calling for it, and if so, what we might recommend overall. So So the purpose really was to focus on that. And we know that since this group began in July, and so again, a very rapid timeline, Jeff Cummings and a group of others published on appropriate use criteria. And since then, there have been many other commentaries, articles, and media coverage, a lot of debate in the field. But again, we were asked specifically to weigh in as the ADRC network. So we approached this by establishing a subcommittee of the CTF to address this. Again, a guiding principle was include every ADRC. And so there was a call to all of your ADRCs asking for a person to be nominated to join this task force. We did want to protect against any conflict of interest bias, but also not work to exclude people that had relevant expertise and the best knowledge. And I think we were successful at doing that. And then, as often in the field, when asked to establish any kind of consensus, when we don't have all the data, we used the Delphi process, which involves a series of anonymous surveys followed by discussion as we vet by a group, and then revisit many of the questions where consensus was not found. So we ended up having 3 rounds of the Delphi surveys and 2 major discussion sessions. And one of the primary issues that came out, 2 major issues really, was, where we need to weigh in and where this is most important is safety as Ajikanamab is being rolled out, and what can we contribute as researchers in a post-Aducanumab world. We're really seen as the areas where the ADRC network can make its greatest impact. And we had several areas of active discussion, and we'll go through these today. The selection of appropriate patients, this question of underserved or non-represented patients in the Engage and Emerge trials, amyloid eligibility, safety monitoring, duration of treatment, educational materials, and provision, not widely talked about, but several active efforts, not just within the ADRC, but in other affiliated organizations. And thinking about how we might be able to leverage the ADRC network and our research experience to really promote further understanding. And briefly, we touched on third-party payer guidance. So there were several major points that really struck me in the discussion. And really, it was an honor to have the group assembled that we had. Papers were flying, points of view, as you can imagine, overall. And it was well recognized that, going back and potentially relying solely on the criteria for inclusion, exclusion, and for how a medication was administered is not really practical once we see approval. Allan Schneider published years ago that if we had done so with donepezil, almost nobody would be eligible for a cholinesterase inhibitor. And again, a recent JAMA article out just a few weeks ago showed that only 9 to 14% of patients with MCI or early AD would even be eligible for Ajikanamab. And I think that is food for thought, not yet a recommendation for us, but food for thought. And then when we think about duration, one of the areas where we struggled the most, and again, we'll show you that data, duration of treatment for most drugs is not limited by the short duration of clinical trials. We know clinical trials are done for efficiency purposes to prove a point. It's really the post-marketing phase that helps us to understand what is the duration of efficacy and a proposed potential duration of appropriate use. Also, with many imaging experts online and as part of the group, clearly understood that amyloid buildup in the brain and eligibility could be determined by both CSF and A beta PET. It's really only A beta PET that can tell us if the amyloid is truly gone from the brain. So those are just some eye openers for me. And we moved into the Delphi and round one anyway. And we can look at, I'm not going to go through these individual graphs, but I just want you to know where we started, with a tremendous amount of debate in the field, bars all over the board, very little consensus overall. And so we moved further into Delphi round two, which I was delighted was more a peaceful protest perhaps. And in Delphi round three, really negotiation, which included points where we agreed and came to consensus, and points where the consensus was that we will agree to disagree on particular issues. So what did we agree on? Well, overall, the committee agreed on patient criteria, general consensus that for right now, under the conditional approval, which is asking for additional study to be done in regards to potential clinical benefit or not for this agent, that the safest and best way to approach this was to look back at the criteria from Engage and Emerge. And this is very similar to the approach used by Jeff Cummings and the group. And those criteria are laid out. Again, I'm not going to go into those in great detail as we'll talk about some of these individual topics as we move along. There's a big question on patients from underserved populations that were not represented in Engage and Emerge. And the majority felt that, yes, we need to use cautions because some of those underserved populations may have inherent vascular or other risks that may alter safety profiles, but that there was no compelling scientific argument to say because underserved populations were underrepresented in Engage and Emerge, that for any reason other than safety, anyone should be excluded from potential access to this medicine. The group as well, I always loved it when we had 100% consensus, and this is one of them. The current conditional approval, accelerated approval for Ajikanamab is based on data showing that amyloid plaques could be removed from the brain, and on the basis of the FDA, a presumption that this was sufficient to suggest a probable clinical benefit. And so this really is a drug that's not appropriate for those that do not have a buildup of amyloid in their brain. That absolutely we have to screen with MRI for safety. ARIA-E and ARIA-H, critically important. And in the FDA label, certainly recommendations for monitoring at approximately a 6 and 12-month time point. There was general agreement that most cases of ARIA occur early on, although they can occur at any time in the disease course. And so a 3-month MRI was optimal. Also, consideration of MRI safety monitoring, that if treatment extends beyond 18 months, it is a good idea to get an MRI at 6-month intervals for safety monitoring. So again, a lot of consensus. So clinically, yes, the group felt as well overall that we needed to continue to monitor people clinically to help with research, to help prescribers and patients and caregivers understand what potential impact of the treatment might be. And some discussion on standardized instruments, looking back at the Engage and Emerge studies, what might be practical in the community. And this was an area when it came to specific clinical measures or standardized instruments where we had to agree to disagree and not reach consensus. Really, the biggies were MMSE, MoCA, CDR, really a split group by perhaps the neuropsychology cognitive group can help us sort through this in the future. Again, these would be guidances, not mandates for anyone out there to follow. So we also looked at duration of treatment. And here, you can see the data from Delphi round 1, Delphi round 2, and Delphi round 3. And yes, our discussions and surveys move the bars a little bit, but at the end of the day, while there was some shifting and jockeying on the field, there was no recommendation that came out a clear winner. The top 3 recommendations were really split decisions, whether or not one chooses to stop at 18 months, whether or not one chooses to recheck amyloid status and determine whether or not it's worthwhile continuing, or whether or not we just leave it up to the treating patient and provider, which is really a default setting. Unfortunately, I don't think, if we did 2,000 rounds of the Delphi, that at the current state of knowledge that we would get any closer to an overall consensus here. We're going to have to figure this out in the post-marketing phase. There was some discussion of educational needs. And so, what are folks going to need out there to appropriately deliver and monitor for safety? And there were 3 main areas that were discussed in great detail where there seemed to be general consensus that there's going to need to be training on A-beta PET interpretation so that people can make that judgment call. Is this patient suitable for Ajikanamab therapy? That radiologists need to be trained in ARIA detection, as do prescribers. In most of our clinical studies, there are both local as well as central reads, and this is really a team effort for safety's sake. And again, prescribers, appropriate use and oversight for safety. I was delighted to see that at least the ADRCs are looking at prescribers being restricted to those with sufficient knowledge to be able to select the right patients and monitor safety appropriately. But what about guidances or recommendations for others out there in the field? We weren't asked to put this together just to discuss amongst the ADRCs, but to provide guidance given the combined expertise and knowledge of the ADRC network. And so educational materials are likely needed. Some debate and no real resolution on these topics. Who should be educated and who should be responsible for that education. So it was overall agreed that clinicians and potential prescribers need to be educated, and that may be our top focus, but patients and caregivers need education as well, and radiologists. A question of who should educate, really a 50-50 tie over should the ADRCs take this on or should we just leave it to other initiatives? So the American Academy of Neurology, for instance, has set up an educational development committee that I serve on and several other members of ADRCs serve on that are trying to put together educational materials. Shall we let them run with the ball, or do we have a part to play? And some discussion on the agreed group led by Neelam Agarwal as to whether or not agreed that is putting together patient-facing materials for amyloid disclosure in cognitively normal individuals, as to whether or not agreed may want to play a role here, or whether the CTF should continue to move forward. So no clear answer yet, but clearly a call for educational material development. And what about research? So we know that many of the questions and many of the issues that were debated in this Delphi process, we simply lack good data for currently. And can we help with that? We are a powerhouse in ADRD research. And so what can we do? And we asked several questions. The two most relevant is if there are folks within our NACC cohorts that receive Aducanumab, is the UDS sufficient to capture safety outcomes and or cognitive outcomes? And the answer was yes, for the most part, but there was really a resounding, we really need to enhance to capture ARIA-E and ARIA-H and maybe other potential side effects for these individuals. And that might lead to our next discussions overall. And then a question of whether or not we could potentially help with the research by trying to recruit folks that are placed on Ajikanamab to be followed within NACC. Overall, the majority said, no, let's not, skew our cohorts by reaching out and targeting recruitment of those on Ajikanamab. We did discuss briefly, and it's certainly not our responsibility or ability to dictate what third-party payers do, but it was felt that important by the group that this should not be a potential treatment, albeit still benefit in question, that should be restricted to those with means, and that third-party payers and their decisions really need to take into consideration fair and equitable access to Aducanumab for everyone. And also a strong consideration that A-beta PET may need to be supported by third-party payers to most effectively, safely, and carefully move into a post-Aducanumab conditional approval world. So what are our next steps? Well, next steps are potential publication of these consensus recommendations, many of our findings redundant to what Jeff Cummings and his group put forward, but some additional commentary and certainly potential roles for the ADRC network in the development of educational materials, perhaps leaning on our ORCORs, who are the experts in this area. How we maximize collection from ADRCs for any potential NACC participants that may end up on Aducanumab could still be a very powerful way to advance research in this post-marketing phase. And there may be many other thoughts. How can we help drive the field forward? And I don't know what the answers are there, but if we can't do it, I don't think anybody can. And so I do think while I was a little hesitant to embark on this journey, I did think that we were in a position by virtue of our expertise where we need to try. We need to try to help, to come together, to provide whatever we can to help everyone out there move forward in this phase of Alzheimer's disease therapeutic discovery. With that, I'd like to turn to a really important discussion, and then perhaps if there are questions, we can get back to them after the fact. But a lot of work by the CTF has been ongoing, led by Suzanne Schindler, Teresa Gomez-Isla, and Jeff Burns on how we potentially capture relevant information on not just Aducanumab, but on many of the other potential disease modifying agents that many of our NACC participants may be receiving either currently or in the future. So with that, I believe that discussion is going to be led by Suzanne Schindler from Washington University. Suzanne.
Greg, thank you so much for that wonderful presentation. All right. So the approval of Aducanumab was kind of a curveball, I think, for many of us. And it's really wonderful that all the ADRCs were able to come together and figure out some recommendations for how to deal with that. And I really congratulate Greg and everyone who participated in the Aducanumab working group for working together to figure that out. So now we're hoping to get some more input into another more narrow topic, which is how do we collect information on these drugs as part of the uniform data set. And we've had a number of discussions about this in the clinical task force. And I'm just going to mostly present some of the major questions we have. I don't think we've come up with any clear recommendations yet, but we're certainly considering some different possibilities and we'd like your input. So 3 of us that worked on this were Jeff Burns, Teresa, Gomez-Isla, and myself. So as Greg talked about, Aducanumab has a major effect on AD biomarkers. We don't know necessarily how much it affects cognition, but it clearly affects AD biomarkers. And those effects are related to the dose and the duration of treatment. And so we think those variables are very important to collect. And there's a variety of different ways that we could collect that information. The first suggestion was pretty obvious. We just use the current medication form. However, I think that some of us had some concerns that this form may not be the best way to capture some of this information. So one issue is that with this drug form, oftentimes people do not include information on when drugs were started and stopped. That's part of the form, but often those fields are not completed. And we think that that time information is very important for interpreting the effects of Aducanumab. Another issue is that the medication form is really intended to be a snapshot, and it includes only the medications that the participant was taking at the time of the visit. So if, for example, the participant was started on Aducanumab and maybe they took it for 6 months and discontinued it for whatever reason, then that wouldn't even be recorded in our current framework. And then one additional issue is that we think it's important to include information on adverse events, the ARIA that Greg was discussing. And where do we put this? So you could imagine putting it in the medical history form. However, a lot of the side effects that are sometimes seen with symptomatic ARIA are pretty mild, and participants may not think to include those when they're discussing their medical history. However, if this is included at the same time that we're asking about drugs like Aducanumab, we may be more successful in getting accurate information. So we have thought about 2 general approaches. So one is adding a section to the existing medication form that includes this information. And then another thought is adding a new or separate form. And of course, we never really like to add new forms, but that may be a possibility. So what would be in this section or form? So the first question is, has the participant ever been prescribed a treatment expected to modify Alzheimer's disease biomarkers like Aducanumab as part of their clinical care? If the answer is no, then that section or form is completed. But if it's yes, then we would want to know the drug name, the highest dose, the start date, and the end date. And then the next question would be, have they experienced any adverse events related to these treatments? Yes, no, or unknown. And then if they have had a reaction, what that reaction or event was, as well as the timing. Now, another really big issue is, should we add more information, not just on clinically prescribed drugs, but clinical trial participation? And this brought up all sorts of issues and conversation. But again, kind of the basic question is, has a participant ever been enrolled in a clinical trial for treatment of Alzheimer's disease? Yes, no, or unknown. And if yes, what is the category of treatment studied? Does a participant know whether they received active drug or a placebo, or were they blinded? And then what were the dates of participation? One thing that I would just note about both of these things, both the clinically prescribed drugs as well as the clinical trial participation, is we expect that both of these are going to become larger and larger issues in our centers. And so that may require more flexibility. So these are areas where we may need more changes over the coming years. And so that's something to think about as we consider how to best incorporate this information into our forms. So at this point, I'd really like to open it to discussion. And of course, Jeff and Teresa can provide their input. So the first question is really simply, should we include information about Aducanumab in the medication form, or should we create a brand new form with this? And then the second question is should we create a new form on clinical trial participation? I would note that this the second item could actually get pretty complicated pretty fast. And so we may need to think more about the best way to develop a form like this. So it looks like there's quite a few questions in the chat. A lot of these seem to be related to basketball. But yeah, and Jeff and Teresa, please feel free to come in. So one issue, possibly collect current dose in addition to max dose. So one thing that we had discussed was, should we go so far as to add the exact dates of dosing and the doses? And we had decided that that would probably be too much burden. I think there's pluses and minuses to doing that. But you could certainly add more information about dosing and times. Can enter clinical trial number rather than categorized drug class. Many are not clearly anti-amyloid. Yes, one issue with that is we are presumably going to be getting some of this information from participants, and they may or may not have that level of detail. Jeff and Teresa, do you want to jump in and answer some of the questions?
Sure. Hi. Yeah, I'd just add, I think the question about adding the clinical trial number, that's come up, and there was some pushback on that for legal or, PHI, I guess, information. Maybe Greg, maybe you want to comment on that, but that's something we thought about, but we're a little worried about the information, legal and, protected information.
Yeah, I have some concerns about the legal contracting for ongoing and even completed clinical trials that frequently have stipulations as to ownership of that data. And so even though we have access to that information, we don't own it, and our ability to share it may be somewhat limited. I think that's something that legal experts are going to need to work out potentially at individual institutions even. And I hope that there's a path forward, because the importance of understanding biomarkers and the developmental work that we're all doing with biomarkers could be severely impacted in a very negative way if we're including within the groups of subjects that we're studying those that have had disease-modifying therapies, irrespective of clinical benefit. If they're altering biomarker outcomes, we need that information. So how we navigate that, those areas of legality, contractual obligations, so that we all don't get sued, is important while we maximize the research moving forward. I don't have an answer though, Jeff, but
Yeah.
I do think I'm hearing a call for an optional UDS form on basketball.
Yeah. Sorry about Massachusetts. Sorry about that. So I do, I think it's great to hear some feedback saying, we need to collect this. And when we're always worried about burden. And so, max dose versus, every dose, we could go all the way to every dose, which could potentially be important if we want real, strong data on exposure. But so sort of what's the tolerance for burden on collecting this is one type of feedback we're looking for.
There are two comments in the chat that I think are very important by Nina and Gil, meaning that referring to how important is collecting whether someone is in a clinical trial, as that relates to the desire to get more autopsies from former clinical trial participants. So that's critical for the field, I think.
And I will throw out there on the GUID front, this is a great concept, but GUIDs are really only effective if everybody uses them, including every trial. And as of yet, unless the FDA mandated GUIDs for all US approved trials, I don't think that the pharmaceutical industry is going to jump right on board. I could be wrong, but I think at this point, GUIDs are not quite ready to capture everything that folks are exposed to.
So just on a big picture level, if we have, say that we have a form on whether or not someone's taking a prescribed medication for Alzheimer's disease, like Aducanumab or other drugs as they become available. And there's another form for clinical trial participation. Should these forms be mandatory or should they be optional?
Would you like people to put their answers in the chat to that?
Sure. I think that one person suggested an optional form, Will Affleck Ash. And then I think Gil sounded, and maybe he can certainly clarify more, but maybe a little bit more enthusiastic in terms of that he thinks would be a great addition to collect information on clinical trial participation. So I think this data is really interesting. It's just, I think that we need people to want to do this either as an optional or mandatory form. And so really we're just asking guidance, like how should we do this? So Hiroko Daj says, I believe it has to be mandatory. And Jim Law is asking about emerging standardization for EMRs to allow detailed medication tracking. I think that's something that we might need to address later. I don't think we're quite at that level where we could rely on that. And then Sudha says initially optional, which I guess brings up the idea that we could start off potentially require or having it as an optional form and then moving it to a mandatory form. Greg, what is your view on whether this should be optional or mandatory?
You know, I think we should strongly encourage all centers that are aware that a person is on a disease-modifying therapy to include that data so that samples can be flagged to not include. We're not going to know in an active clinical trial if they're on placebo or the real thing, but we could know this person's biomarker data may not be reliable, may have been altered in some way. And I think that's critical. And that's why I wonder whether or not we can get around some of the legality issues by simply describing the class of drug. Were they on an anti-amyloid therapy? And that way we're not disclosing a Biogen specific or an Eisai specific or a Roche specific drug. But we know they're on an anti-amyloid therapy. An anti-tau therapy, an anti-inflammatory therapy. It would give us some way to figure this out. I hope that we can pull together. I'd like to see this eventually as a mandatory form for those who can be identified as having been on a disease-modifying therapy. But again, you know, the CTF doesn't do anything without consulting with all ADRCs. So it's really, really important that everybody contribute here. And certainly, I would imagine Suzanne and Teresa and Jeff, that the plan is, as a form starts to become developed, that this will be shared as we have our other forms with the ADRC network, including all sites, for input and further discussion on this just critical topic for the field.
Yeah, I certainly agree with that, Greg. So just a couple other comments. So Suzanne Hunt says, if the form is optional, you can only use the data where people report drugs. Unreported drugs doesn't mean no to drugs. So that seems to be an argument for making it mandatory. Of course, you know, we're going to have to include Aducanumab in some way. It's just the level of detail, really. And then Charles DeCarli says, I think the duration of all medication use should be tracked. We have developed a form that gets at this in a broad way for vascular risk factor treatments. So yes, I think duration of treatment is an important variable. I think it's more important for some drugs than others, and for Aducanumab, it's incredibly important. Again, I think it's a level of detail sort of issue. And is this lumped together in another form, or is this a separate form? And I think there's kind of pros and cons to both. I think the biggest issue is we just don't want to increase burden for sites too much, or to an extent they don't find it acceptable. Teresa or Jeff, did you have any additional comments?
I'll just make one last comment. I mean, I think it's important to remember we're doing two things. One's probably mandatory. It's if they're prescribed a drug, we should be recording it. And then the question we're asking is how much information should we record beyond, you know, just drug name. And then the other is the clinical trial participation, which I think is related, but a different question. So, and the feedback you're giving is great. It sort of encourages us to keep moving on the path. And we will, yeah. So we'll pilot. We'll give you a taste of it and get more feedback as we go.
I agree with Jeff. We definitely need more feedback. Somebody was saying that perhaps the chat is not the ideal place to just vote, and that's not what we are aiming at here. Just to get a first sense and then get feedback and pilot a form.
That's great. If there's any final discussion points, we've got about 3 minutes if we want to give that. Otherwise, we can wrap up and move on to the next item too.
And I'd just like to encourage everybody out there at the ADRCs, you know, if you have a real interest and expertise in this area, to reach out to Suzanne, to Jeff and Teresa, and offer to lend your particular expertise. The CTF itself is dynamic, and not just, as Allan said, are we building a dynamic UDS 4.0. We bring in expertise when expertise is needed. And clearly, there's a call here for doing something and gathering and using our network resources optimally is critically important. Not to inundate you, Suzanne, Jeff, and Teresa, with email offers, but I do think important. Everyone has a voice.
Yeah, we could definitely use some help with this.
Yes. And somebody, I think is from Dr. Mesulam's Center, says the chat is definitely not the format for this discussion. We agree. So a lot more to discuss.
Okay, well, thanks, Sienna. I think we're ready to move on.
All right, well, up next we have got both Greg and Cindy presenting on the UDS4 revisions. They are members of the Clinical Measures and Diagnosis Subcommittee. I did want to also point out, I've been sharing in the chat a link which has all of the Zoom links on it, but then if you scroll a little bit farther down, there's also links to all of the forms that we'll be reviewing today. And so you can open those if you have a double monitor, especially. It will be helpful to have those open side by side if you'd like to follow along with the forms for today's discussion. But go ahead and take it away, Greg, or Cindy, whichever one of you is starting.
Fantastic. I think I'm going to begin, and then we'll switch over to Cindy, as everybody is probably sick of hearing from me. So as you all know, and as Allan pointed out, the CTF divided into sub-working groups. And so the Clinical Measures Diagnostic Working Group, chaired by Cindy and myself, with Leanna, Brad, Jeff, Teresa, and Howie, have been working on the forms that I've listed below. And I really do want to thank NIA, Nina, Cerise, and Grayson for all of their help and support with this, and a real call out to Bud and Jessica and Amy and Hannah from NACC that have been incredibly responsive, supportive of this effort as we move forward. So again, a team effort, again, coming back to what Allan commented on earlier, key guiding principles are, number one, best science first. But, you know, a strong call to see what we can do to streamline the battery as much as possible to reduce subject and site burden, allow increased harmonization, and dynamic adaptation. And we're not saying, you know, that UDS 4.0 is everything. We know that sites all have unique measures that are driving their own science, but that this core is credibly important. And we do hope that the dynamic nature of UDS 4 really encourages groups of centers to move forward with uniform collection in areas. So again, you know, we're working on that nucleus of the atomic atom, and one can see how that might fit with different content. You know, we have a DLB battery, an FTD battery. There are site-specific measures, whether or not we develop an experimental drug form, whether or not additional information is needed for co-participants or social determinants of health, or issues that might be optional forms in addition to UDS 4.0, but UDS 4.0 really a core harmonization tool for all of us. And I want you to know, that, and I speak on behalf of the entire CTF, this is not a group of people in the dark working on this. We have been calling out to you with multiple surveys, probably too many. Your site's probably sick of hearing from us. Focus at the spring ADRC meeting with breakout rooms that were incredibly helpful for us. And the CTF was incredibly responsive to those suggestions. And then, of course, the exercise that we've gone through over the summer with formal center feedback. And I want you to know that the group has really taken great pains to incorporate all of these comments, especially the comments that have come from large numbers of centers, or even those that have come from even a single center if the scientific argument behind them was strong enough over time. And again, as the center network, all of our sites, we need to continue to work very closely with NIA and NACC to make sure we're taking into account all areas that could be incorporated in a core battery to promote scientific discovery, and again, working with NACC to make this practical and doable for individual centers. So there are a few principles that really spread across all forms that we're going to present today. And so we're going to really cover these here all at once. So there was a strong call across many of the forms to be consistent in our use of semantics, our phraseology. Use participant, not subject. Use co-participant, not informant or study partner. There was also a great deal of debate across forms on timed events from the medical history or from symptoms or for diagnosis, whether or not, in the UDS-3, sometimes age was used, sometimes year was used. What is best? And so the committee has debated this extensively and decided that most report from participants is probably most accurate when you ask them at what age. If somebody asks me at what age did I have an appendectomy, I can tell them it was age 22. I have to back calculate if people want to know what year that is. And that's not a major issue, but it helps for us to standardize. A lot of questions on initial visit versus follow-up forms. The group is really thinking that the same forms probably should be used, but we should allow a carry forward of responses and then instructions for sites to correct if new information becomes available. They told me last year that symptom onset was at age 72, and now this year, I've been able to rifle through the electronic medical record and find that that was really 2 years earlier than what was previously reported, and or if new symptoms or medical conditions emerge, these forms could be updated as well. And remember, that it's not just about the questions in the UDS battery. A lot of the revisions that we're putting together or instructions to harmonize completement of the forms. And we'll talk more about that as we go through the forms themselves. So again, these are the forms, and we think we have these in really a finalized state or very close to anyway. And the major strategies we've taken with a one participant demographic, streamline the old and add new items, occupation, ADI, some social determinants of health, co-participant information, refocus on the issues of contact, reliability, and limit demographics for centers that are interested in co-participants. We encourage those centers to get together and develop an optional form that could be used across interested centers. Family history and genetics, really leveraging the concrete data coming out of ADGC rather than a simple report. Medications, update this list, add Ajikanamab, certainly link to RX norm in a REDCap based system as well. Medical history, we used to have A5D2. Let's have a single form focused on best data and update items. Physical exam, neurologic exam, clinician judgment of symptoms, all very, very important to streamline, to bring our science up to date in terms of many of the areas of active research. And then for diagnosis, really, as we are in an era of biomarker development and increasing use of biomarkers across the ADRCs, really incorporate this, but don't diminish or exclude the clinical diagnosis as well. So we'll go over these individually as well. There are some other forms in process, as we've heard about, an experimental medication form. This is kind of being worked on currently. A potential expanded social determinants of health form as an optional form for centers that may be interested. And we're not going to discuss those other forms because they're works in progress today, but certainly, as we said in the last session, everybody's going to be brought on board to weigh in if they have an interest. So we're going to start with the forms, and the format for our presentation is really a very simple. In each form, we're going to go through this in the same way. I encourage you all, NACC has put the forms on that Fall ADRC landing page, and encourage those of you that are two screeners to bring those forms up and follow along. But our slides will be focused on what we heard. What were the ADRC comments, the major comments? What proposed changes did we make on the basis of those comments? And if there were comments that were not incorporated, why that decision was potentially made. So very, very important. We don't have any hard timelines. We're trying to move this forward as rapidly as possible. But again, guiding principles are best science first and harmonization. So we'll keep you all updated. I know that many of the database managers and other folks at ADRCs are saying, when do we need to implement this? We'll keep you posted as we get closer to any concrete dates or implementation phases. So we'll start with Form A1. This is the participant form. There were several suggested changes asking about gender identity, not just sex at birth, clarification of primary language. We had deleted the handedness item, but there were several centers that wanted that kept. And there were several centers that commented on the burden of looking up occupation as well, and a new item really coming out of some of the discussions at both the spring ADRC and AAIC this year as to adding the area deprivation index. So there were multiple questions or comments on social determinants of health and or lifestyle questions, including people in opt-out button, use household rather than personal income, and then for some of the social determinants of health, some suggestions for alternative items that were put into play. We'll talk about that. Folks looking when it comes to contact with others, including some of the more common ways to maintain contact in our current state of social isolation, text, social media, email. Critiques that self-report was unreliable, although how we get around this is complicated, and many centers looking at for lifestyle factors, quantitation of time and intensity. So here's what we did based on what we heard from you. We added a question on present gender identification. It'll be helpful, and we do have a session on LGBTQ in the OR session this afternoon. Hopefully we'll be able to help with that with this addition. We've provided clarification on primary language in question, and if sites that have a real interest in this issue of primary and secondary languages, we'd encourage you again to work with other centers that may have shared interests to pull that together. We put the handedness question back in. Again, we're working with NACC and to hopefully develop a strategy for very simple codes for occupation, much like we use on the current medication list. If we have a medication that's not in the top 100, we type in the name and it pops up a code. We input the code. The same is going to hold true for the new question, looking at ADI, and we're really looking at, ADI information. What's the state decile? What state is a person from, and what's the national percentage? This is going to be a lookup item again, but hopefully with the help of NACC, this is going to be a type in their zip code. You don't have to share their full zip code if that is not IRB permitted at your site, and transcribe this information. An additional new question was put in on military service. This is important for some of the ongoing NIH and NIA initiatives, and this is a simple, again, using form logic. If the person was in the military, there may be an additional question. If they weren't in the military, it's a simple checkbox no, and you move on. We did change the social determinants of health mini battery. Again, just a few questions, 5 questions covering 5 core principles of social determinants of health, allowing people an opt-out for all of that information. And many of the items have been modified to optimize the feedback that we've heard from you all today. So what did we not integrate in the form? Again, there were some comments that appeared very idiosyncratic, site-specific. We encouraged the sites to continue to collect your site-specific measures, and that those are best handled at the site level. Again, we're going to work closely with NACC to try to facilitate and make it as easy as possible for any of the coding lookup for occupation and or ADI. And again, there's ongoing work on an expanded social determinants of health battery. So Form A2, co-participant form, we had streamlined this to take the focus off the co-participant to focus more on their reliability to report on the participant themselves. There were 4 centers that clearly requested that we keep detailed co-participant demographics. Many other centers commented, more centers commented on appreciating the brevity of the form. We would encourage those 4 centers, and I know here at UK, we have a very, very detailed co-participant packet, which is ancillary site specific. It'd be nice if we could come up with a uniform optional expanded co-participant form for sites to choose to use if they have that interest. interest, but not to overburden sites by putting this within the core. There were several sites that noted the importance of texting email again, and several centers commented. The prior questions had talked about interactions at the 1 hour per interaction stage. And many folks, I think, rightly commented that sometimes we may have caregivers, that swing by in the morning for 10, 15 minutes, swing by in the afternoon or evening after work for 10 to 15 minutes, and are providing supervision, care, and observation during those time periods. And we thought that was an important consideration. So the changes that we put in play were to allow these other modalities as answer choices in terms of how that co-participant is gathering their information, their contacts, and to take into account contacts shorter than even 5 minutes, such as text or email as well. And this will help us actually determine absolute contact time. There could still be some debate about quality of contact, but we tried to do our best to be responsive. So there were some changes, again, that were recommended from a limited number of centers. Again, we encourage centers to work together. We did not want to overburden that core UDS4 overall. So A3, our family history and genetics form. And many centers had comments. There didn't appear to be any major overarching issues or consistency, but these were important for us to consider. Understanding whether or not there's a genetic mutation in the family, important because we're not collecting blood or getting ADGC to perform analyses on family members. What we do about ApoE status, potentially removing birth year, because sometimes, people don't necessarily remember exactly what year, and instead allow perhaps some estimates of age for at death or year of birth. Provide options of adding more than one condition, create subcategories of psychiatric conditions, require information only on siblings if they had a neurologic or psychiatric problem, and then thinking about broadening the categories to include different categories of Lewy body disease, PPA, behavioral variant, FTD, PSP, vascular cognitive impairment and dementia, and cerebral amyloid angiopathy. So what did we do? Well, overall, we felt that trying to keep the birth year for siblings, but allowing estimates for that birth year, age of onset, and age of death. And this is all really provided in instructions. It helps us to understand that familial occurrence considered important. Potentially think about reporting detailed information only on those with neurologic and psychiatric conditions, but it was felt important to understand the number of siblings so that we could place that in context and understand the genetics. Certainly expanding categories to add subtypes of FTLD, and to use the other response form to specify for less common conditions with less genetic weight or information. So there were a few suggestions not integrated into Form A3. Keep the questions based on best diagnosis because report of mutations was considered potentially inaccurate. And I will say I agree with this. I had a patient yesterday who informed me that they had an autosomal dominant mutation for Alzheimer's disease. And after digging through the information, it turned out it was ApoE, not a presenilin or APP mutation. Use the NACC ApoE rather than relying on self-report. We should be able to trust that data. I trust that data. Limit to primary diagnosis. Secondary diagnoses may just complicate things for siblings and relatives. And a thought that expanded list of psychiatric conditions, again, may just make the form more cumbersome for those that are very interested in independent psychiatric conditions. We again would encourage sites to work together. Again, our general principles for what we didn't change. UDS Form 4 for medications. A lot of centers called out and said, you know, we're not ready for EDC or REDCap RX norm. We need to make sure that we have a paper form as well. And we understand that. And certainly we think that that's important. The practical implementation is critical. Several centers commented on the need to specifically capture Ajikanamab and other biomarker modifying therapies, as we've discussed earlier today, and really to update the list of medications from UDS3 to what's being used now in terms of other medications, new anticoagulants, new type 2 diabetes medications, more commonly used medications, including dietary supplements. So what did we do? We had decided that both EDC, REDCap, and paper forms should be viable methods for collecting to give sites flexibility. That Ajikanamab as a prescribed agent is certainly going to be added to the formulary list. The development, the work again that Suzanne, Teresa, and Jeff are doing currently on an optional form or mandatory form for disease modifying therapies. And again, a formulary list of medicines being updated in NACC and RX norm. So really, we think we were successful in really incorporating all of the recommendations in Form A4. And with that, I am going to mute myself and introduce Cindy Carlson, my co-chair for the Clinical Measures and Diagnosis Work Group, to move on with our additional forms. And Cindy, I'll advance the slides for you.
Great, thank you, Greg. So again, thank you all for your input on these forms. I know it took a lot of effort to gather your centers together and gather some consensus input, but we really appreciate it. I want to thank Greg and the CTF members for the clinical diagnosis group as well. So we'll mention their names as we go along. So Suzanne Schindler led this team. And we discussed, this is the form, the combination of the medical history form. So again, as we discussed at the spring meeting, the medical history form previously was broken up into the A5 and D2 forms. So these have been combined to streamline the information collected on the person's medical history. And so some of the suggested comments that people made across the centers, looking at the consistency and age of diagnosis. So like we mentioned, using year versus age. And as Greg mentioned, we settled on age, thinking it'd be most consistent for the participants, and then consistency in answer choices. So again, across all the forms, we'll be looking to make sure that the answers are as consistent as we can. There is some input on clarifying the time frame for alcohol use and rewording some of the alcohol questions, and then changing street drugs to recreational drugs, adding frequency of cannabis use. Some clarification just to make sure that as coordinators are reading questions or people are asking participants, if they've had a heart attack or cardiac arrest, they use lay terms to help them understand what that was, so that they don't inappropriately say, oh yes, I had atrial fibrillation episode, and have that listed as a heart attack. So there's a lot of input on the details and activities related to head injuries, some input on type of cancer treatment, and then clarifying details of obstructive sleep apnea treatment. Next slide. So the proposed changes that we'd like to make based on the ADRC feedback, so clarify if smoking, alcohol, or other substance use is absent or unknown. So sometimes the questions would say absent, unknown together, and then you're missing out that someone didn't, or they just didn't ask or weren't sure. So clarifying that. And then in the alcohol use questions, really clarifying, so in the time frame, clarifying the time frame, and then on the day that people do drink alcohol, how many would they consume? So just kind of clarifying some of these details so they're more precise, and then also adding in standards for alcoholic drinks so that the coordinators, the clinicians can have that readily available and be consistent across centers. Next slide. And then just clarifying again, prescription or recreational drugs as far as using substances out of prescription, and then clarifying again the time frame within the last 12 months or prior to the last 12 months. And then again, refining some choices of both substance use and alcohol consumption, reframing some of those time frames as was recommended to never, a few times a month, a few times a week, or more. So having it be consistent across substance use and alcohol use. Next slide. So again, these are the changes. So if you, again, keep looking at the forms, and I know there were some comments in chat and questions that some of the forms don't have all the precise details, and they're still in process. So we will get those details addressed as have been raised. NACC has been amazing at helping us work on these forms and update them, but a lot of these discussions have been going on till the weeks before. So, so we will get those changes made. So again, just some specifics here on clarifying whether something's active, remote, clarifying language instead of speech, clarifying the timing of carotid artery surgery and the age of most recent surgery, and then terminology such as other Parkinsonism disorders, and as has been done in other forms as well. So next slide. And then there's a lot of comment again on the head injury questions. And so those were refined, and again, with a lot of input from a lot of you who have expertise in this area. So kind of redefining the periods of time. So that critical cut point of less than 5 minutes, 5 to 30 or more than 30. And then head injuries, describing more of a concussive symptom, so feeling dazed, confused, seeing stars, and then capturing other kind of repeated head injuries. And as you'll see on the draft forms, capturing other activities that may have been related to head injuries, repetitive, subconcussive injuries. Next slide, I mean. And then clarifying with obstructive sleep apnea, again, for CPAP usage, to clarify the number of hours per night. So as opposed to 50% of the night, instead to have how many hours used per night. So 4 hours, less than that, more than that, etc. And then if somebody used an oral device for sleep apnea with the same kind of time frame used to describe that. Next slide. And then clarification of types of cancer treatments, again, recognizing there's some questions about chemo brain or other impacts of surgery, etc. So being able to capture that a little better, and then leaving options for other medical conditions or procedures with free text to be able to add in. So again, we tried to streamline this, but leave open for other conditions that maybe become more prominent later, things like COVID-related cognitive changes. So as those conditions become identified and named and clarified with criteria, that we would be able to add in some of those conditions. Next slide. So the suggested changes we did not implement, some suggested I think one team suggested breaking the heart failure into high and low ejection fraction, but at this point we didn't think that was that people would really know what their ejection fraction was and the details involved in that. We weren't sure what the yield would be. Similar with focal or generalized seizures, it may be too detailed to capture an unclear scientific value without having to gather more detailed information. Again, some suggested using a brief surgical history and part of it to capture anesthesia. Some key surgeries are already identified, and this would be a great detailed project or maybe a center specific project, but that was probably a little too detailed for the core group of questions. And then the question about hormone replacement therapy, some of that should be captured in the medication list. And then the concern about difficulty in assessing years of menopause, since it can occur over several years. So, all right, next slide. And then other specific conditions, multiple sclerosis, migraine, years in primary sport. So again, there's some good suggestions, but probably just a little too detailed. Again, trying to really keep this as a core battery and not, but again, allowing centers their choice to be able to collect these other questions based on their center's interests. Next slide. So moving on to B1. So B1 previously was the one called physical exam, but was really just the vital signs, anthropometrics. So there were some, we had proposed switching to metrics just for consistency and calculation of BMI and analyses. Some people had concerns about that their scales and their centers may not have metrics. And so others said they would just, gather it in the English system and just convert it automatically. So we thought that was very reasonable. And we'll talk about that when we talk about the changes. Some proposed doing 2 blood pressure readings per arm and then automatically calculating to reduce error, as opposed to having the coordinator calculate the average and then entering that. Many centers supported having a research grade blood pressure, given the importance we were recognizing of blood pressure contributing to cognitive decline. However, some centers also were concerned about that it was going to be not practical to assess blood pressure in this standardized approach. And then other centers wondered if, because the blood pressure module, which we integrated into this, that was developed by colleagues at several centers here, we had integrated that module that was more recently developed into this. They wondered within that module of the type of blood pressure cuff, the specific type, the not using automatic cuff, if that was going to be a challenge. So, all right, and I appreciate the comment that that we need to follow having a standardized approach. So, all right, next slide. So again, within there's also some very nice guidelines about the current recommendations for blood pressure treatment and making sure to give feedback to participants. And so there's some question about, do we need to have this much guidance for blood pressure? So, whether we should simplify that. There was a question about noting time of blood pressure medication, but that was not recommended not be transmitted to NACC. So why are we collecting it then? Concerns that there may not be agreement on blood pressure recommendations, or that they may change over the course of the UDS-4. So, why are we including them? There was somebody raised the point about why aren't we measuring orthostatic blood pressures? And then questions about different blood pressure cuff sizes. Next slide. There are also some concerns about questions whether participants would feel comfortable having waist tip measured, and how research assistants would be properly trained in this. And then one center was concerned about removing the vision and hearing question, which has been moved to the cognitive component to see if vision or hearing was impairing their cognitive performance. Next slide. So what we proposed based on these feedback was to go ahead and keep the measurements in pound inches and then convert from English to metric automatically, since we'll be moving more towards electronic databases. Clarify if the blood pressure was done per the blood pressure protocol. So again, I think many centers recognize the need for a standardized approach to blood pressure measurement as best as we can. And so if a center is not able to do that, that they would just check that they did not follow the blood pressure protocol. Again, having the 2 blood pressure measures averaged electronically, as opposed to having to do a calculation, which is prone to error. And then removing the requirement to use approved blood pressure cuffs. So there was a list of blood pressure cuffs, but training people on the appropriate blood pressure cuff size, being consistent in the use of the blood pressure cuff, a standardized approach to doing that. So at least within the center, it would be standardized, even if it's not standardized across all the centers, recognizing that there are other studies within the center that need to be standardized in their approach to measurement of vascular risk factors in allowing automated cuffs. Next slide. Someone asked about timing of heart rate, so we're just doing that. And then we decided that, because of the importance of metabolic factors in assessing cognitive risk, that we would leave in the measurement of hip, waist-hip ratio, provide some education on how to do this, but then also providing a participant opted-out option. So we've been collecting this at our center and have had very good success rate. We have low rates of patients refusing to do this. I meant to pull up the exact number. I had my team member look into that, but we have that information. And again, it will be an opt-out because some people do feel uncomfortable having that done, but at least for those who are comfortable, that we'd have that information. And then for the proper blood pressure measurement section, we would streamline that, take out some of the features that didn't seem appropriate, but also across all of these provide some better guidance, clear instructions, and training modules so that team members can feel comfortable in how they're gathering the blood pressure and other measures. Next slide. So again, things we did not add back in. So the questions and vision and hearing are already added in the neuropsych forms. We did not add those back in, but again, centers can continue to do this if they find that helpful. And then we thought that adding orthostatic blood pressure measurements would be prone to a lot, probably even greater errors than even just regular blood pressure measurements. So we thought we'd not add that right now, but if a center wants to do that, that would be good pilot data. Next slide. All right, so I know we're going lightning fast. We have a lot to get through, and now we're just getting to the meat of things. So again, keep following along with these forms. And this is the B8, which is the physical exam, called a neurologic exam, but as an internist, I think it's more of a physical exam, neurologic exam as part of that. So again, this is where we really want to make sure that we have good instructions up front, that the header is informative, and we'll talk a little bit later, but trying to have links to how do we define things? How are we consistent in measuring these findings? So in a lot of this across these next forms, these B8, B9, and D1 forms, you'll see is just to document what's being done. Instead of mandating who's going to be the examiner, that you document what are the qualifications. Is it a trainee? Is it a nurse practitioner? Is it a neurologist, geriatrician, et cetera? And then improving instructions, definitions, terminology. So again, helping it to be standardized across just to reflect the state of the science, updates in terminologies, et cetera. So a lot of those I didn't get into in detail, but just to know that we've seen those recommendations and we'll be making sure that the terms are updated. Some recommended grouping physical exams. Oops, if you can go back. Grouping physical exams, some had noted too much detail on rare findings. Some clinicians prefer the old form. And one thing that's really important that we talked about a lot is distinguishing between not assessed and finding not present. So by someone not having something, it was not clear previously if it was not assessed or that they really didn't have that. So we try to clarify that. And then better alignment of just the visualization so it's easy to be filled out appropriately. And then as mentioned, video demonstrations for standardization of physical exams was recommended. Next slide. So again, the more detailed recommendations, so clarifying if it was partial, comprehensive, in person or telehealth or video health, adding an unknown, clarifying the value. Some people raise a question, does it really matter if the participant has been seen by the same clinician previously? What part of the exams were completed to state that there is no abnormal findings? Some recommended having continuing to keep in the right-left orientation is right-left documentation, not just unilateral findings or bilateral findings. And then some people had some more very specific recommendations about physical exam findings in more rare forms of dementia. Several recommended removing Hoehn-Yahr staging since it was chiefly designed for Parkinson's disease. And then people wanted clarification on the additional measures that were listed. Will NACC support those? How will we collect those data, et cetera? So next slide. So the proposed changes, again, were to clarify, is this a comprehensive or focused exam? Is it in-person or virtual? To note the training of the clinician, update the terminology, modify the format so that it's clear, and you can see some of the proposed changes that are, again, in process on the forms as you look through those, and then remove the Hoehn-Yahr staging question. Next slide. Suggestions that were not integrated. So some recommended that the UPDRS be added back in. Again, we decided to leave this as an optional measure. A lot of teams have been collecting this since UDS 2 and through 3, 4. So if centers want to do that, there were some suggested standardized measures. So again, integrating that use across your center if that's what your center's focus is. And then that can be combined with other centers that are also interested in that. And then again, the right and left orientation. So thank you to Brad Beauvais, who led this discussion group, but thinking about use of left-right abnormalities on an individual basis are very helpful. And the individual sites obviously will have access to that information, but it was unclear whether that would be helpful in longitudinal and cross-sectional data analyses in UDS. So again, we've been able to partner with NACC and look at a lot of the various measures that have been used in previous publications. And so there was some uncertain value for each finding to designate whether it's left or right. And then this format was thought to be more consistent with FTLD and LBD modules as well. Next slide. Okay, and other findings, again, some rare findings, some other pathologic findings that are non-specific or captured in other sections were not added into the B8 form. Next slide. So now we're moving on to B9. So again, this is clinical judgment of symptoms. And this is where we're, again, pulling together information from the participant, from the co-participant, and then the clinicians integrating these information. So, again, there were suggestions to reduce redundancy with other forms, specify mode of onset, that we're referring to the predominant symptom. Some other additional symptoms like behavioral change might come up abruptly, but it may have been the cognitive change that started their time course. And then specify more clearly who's providing the information, clarifying when we talk about changes compared to what baseline or to their last exam, being consistent in use of decline changes. And then some people said don't ask the participant about the behavioral symptoms because they may not be aware of them. And then there was a point that they should not have an option for no co-participant being present. Next slide. So we did clarify, so some people said remove names of people from orientation because it's really more of a memory issue. So kind of clarifying a lot of suggestions to clarify the wording to be more accurate and precise, clarifying the timing, adding some terminology that would help better reflect CTE, and then some clarification about whether hallucinations are well-formed or not well-formed. And then again, just questioning whether we should add back in the most prominent change in behavior from the prior. And then removing reference to REM sleep disorder and adding maybe another question about confirmation of REM behavioral disorder. It was one suggestion that somebody had. Next slide. So in B9, again, so other changes, adding peripheral neuropathy in addition to arthritis and injury causing some gait changes, some other specific findings about facial expression, changes in motor function based on stroke, and then people had suggestions about timing and baseline for falls and how do you describe falls. Next slide. So based on that, we propose these changes. So again, for each major domain, that the mode of onset should characterize the predominant symptom that has most affected the participant. So just clarifying the wording for that. specifying use of the term decline instead of just changes. Again, clarifying the precision of the description of, like, such as, names of people, removing that from orientation, changing the prompts for judgment questions, added trouble with speech comprehension, and then clarifying and defining specific wording of cognitive and behavioral symptoms. So a lot of very specific recommendations that were integrated. Next slide. And then changing this one question to specifically ask about dream enactment, and added a sub question about whether REM sleep behavior disorder was confirmed by polysomnography. Adding some questions to cover CTE related behavioral changes, clarifying some timing for depression, adding the peripheral neuropathy, and some separating out facial expression from slowness, and then trying to be more clear about the timing and types of falls and what baseline we're talking about. And then some questions were added about limb weakness and slurred speech to cover stroke symptoms. Next slide. The things we did not integrate into B9 are, there's some redundancy with other forms, but sometimes they're part of a set scale or important elements that kind of align with this section. So again, we really have worked to try to reduce redundancy, but in some cases there will be some questions that are asked several times. And then don't ask participant about behavioral symptoms. And so we said we thought it's still helpful if they're not aware of those behavioral changes. And then allowing more detail about who's providing the information. And again, recognizing that this is a clinical form, so we're assuming that the clinician is pulling together all available information, as is done with most clinical formulations. Next slide. Other things not integrated, we did not add back an UDS-3 question on most prominent behavioral change. We removed unknown from individual symptoms so that, so again, somebody suggested removing unknown, but again, we wanted to really clarify, was this asked or was it completed or was it just unknown? And then trying to improve variability, and then recognizing that there are sometimes visits where a co-participant may not be present. So we just left that in there. Next slide. Final home stretch, and this is the crux of a lot of these questions. So D1A, again, is getting more at the clinical syndrome that's been evaluated, coming to a clinical diagnosis, and D1B is going to summarize more of the biomarker etiology. So some suggested to further separate severity and etiology, simplify the MCI data collection, but also identifying MCI subtype and domains in dementia. Some suggested adding apraxia as a domain for cognitive impairment and adding additional specific syndromes. And then people had questions on what's the purpose of asking, was it neuropsych data used to inform the clinical diagnosis? And then subjective cognitive impairment, and clarifying the cognitive impairment, not MCI questions. People had, suggestions that we should clarify those definitions. Next slide. So for the proposed changes based on these recommendations, one thought was to change, or our thoughts were to change this from 2 sections. So just the cognitive behavioral status and contributing conditions to 3 sections. So level of impairment, clinical syndrome, and then contributing conditions, or the etiology. So we'll have this reflected so we can, again, get the level of impairment, the clinical syndrome, and then the biomarker-based assessments. And then as far as we said, well, we'll, instead of telling people they can't use certain information, or can instead to clarify what information was used to formulate clinical diagnosis. And again, most of us as clinicians thought that most of us will be using all the clinical information that we have, whether that be biomarkers, cognitive data, clinical information, physical exam findings, to formulate clinical diagnoses. So we would just identify which data were used to support this diagnosis. Next. So again, additions, adding apraxia to choice of domains, adding categories to help clarify what cognitive impairment, not MCI is, adding to syndrome choices, a few key areas, but again, several other areas were not included just to keep things more streamlined, deleting probable and possible AD as syndromes, and then clarifying some of the differentiations of TBI from CTE, etc. Next slide. Again, suggestions not integrated. We did not integrate questions about vascular dementia as a clinical syndrome. That would be more into the etiology of the cognitive changes. And then depression, anxiety, people asked for more granularity, and this will be covered in, again, hopefully in B9 and the NPI. And then combine, people asked to combine neurologic, genetic, or infections with different medical conditions. And so we did not integrate some of these changes. And then we do need to still clarify subjective cognitive impairment definition. So these are things not integrated yet. So for D1B, so this is the biomarker section, people suggested adding the timing of biomarker assessments, what's meant by molecular neuropathology, documenting negative biomarkers when they support exclusion of a diagnosis, adding SPECT, CT, removing centers standards for positivity, clarifying, is this going to be completed for all or even those who are cognitively normal? And then clarifications on MRI and ApoE4 status. Next slide. And so based on this, we recommended that the form be changed from biomarker-based diagnostics to biomarkers in etiologic diagnosis, to specify the year of biomarker assessment. So again, age we'll be using for participant self-report, in year because it's easier to look at a form that has the year the MRI was done, etc. And then clarifying molecular neuropathology to refer to amyloid, tau, synuclein, etc. And then where it was drawn from, CSF, plasma, or PET tracer images. And then add checkbox for indicating no biomarkers were collected, and then adding specific things such as atrophy and asymmetric atrophy for MRI findings. Next slide. We're almost there. So again, we did not integrate some other changes such as PET as etiologic biomarker, or stating that it's etiologic biomarker. So with the new form is clarified to be biomarkers and etiological diagnoses. So it suggests that, again, it's not, the diagnosis is not completely made in biomarkers. And then we, again, try to keep things simplified. So there's some suggestions that we didn't add on method of analyses, other details. And then ApoE status, again, will be objectively evaluating that elsewhere. Next slide. So again, for the next steps, again, I know this is a whirlwind. We wanted to kind of touch base on, as Greg mentioned, the approach we took. So again, we really appreciate all of your input. We integrated it. We went through every single word. We integrated most of the findings. Some findings we left for center-specific data collection. And so next steps, we still have to look at methods for collection. So is this something that could be mailed to the participant ahead of time so they can remember when their aunt or when their mom died from whatever conditions, and ponder that with family input, or electronic collection in person. And then instructions for forms. So again, we're trying to work on more detailed instruction in the forms so that people don't have to go to the code book, or else having some embedded links, something to make it more reliable and harmonized. We're proposing some webinar trainings and video library to improve standardization across form completion, other assessments, physical exam findings, and then development of accessory packets based on collective center interests and areas of focus. So if they're interested in vascular area or FTLD, that maybe they'd have accessory packets that they develop to share across centers. There will be a transition to REDCap within NACC, or there is in the process, but Ben Keller is going to be talking about that tomorrow. So we encourage team members to do there, but there will be paper forms available still. So again, while we're moving towards electronic data capture, there will be paper forms available. So final slide. Again, this is a real group effort. So I want to thank everyone in the clinical task force, but in so many hours, and all of you, because all of you have contributed through your centers, collective effort to refine these forms. So really thank you for that.
Fantastic. And I know there's quite a bit in the chat, but we're a few minutes behind schedule. We certainly recorded all of that, and we'll take that into account. I think we should move to our break if I'm not correct, Hannah. And again, you'd please reach out to Cindy and myself with anything pressing on these issues. We know that was a tremendous amount of information. Again, as the forms become modified, we'll be able to look at this. I think there's a lot of concern about the length of UDS form. Remember, we trimmed quite a bit as well. Where you know, the practice or the proof will be in the pudding eventually. But we're hoping that a lot of it has been shifting items to make it more intuitive. And again, to use form logic whenever possible to limit responses. Cindy, anything else?
I think so. I'll try to answer some of the questions in the chat and on the, you can put it in the question format too. We'll try to get back to team members. I know there's a question about the COVID impact surveys, so that we're trying to align with other initiatives across NIH. So that's been being worked on, but wanting to try to align with other NIH initiatives as well.
All right. Well, with that, let's take a now modified to a 5-minute break. And we'll resume everything back here again at 9 a.m. At 9, we'll have the Neuropsychology Subcommittee speaking with Dr. Lisa Barnes starting us off. All right, that was a very quick 5 minutes. Lisa, are you back and ready to resume?
Yes.
Awesome.
Okay, do you see my screen?
We see it in edit mode.
What about now?
And now it's presenter. Great.
Yay. Okay. Okay, good morning, everyone, or good afternoon, wherever you are. I'm Lisa Barnes, and I am one of the co-chairs along with Andy for the Cognitive Working Group. And we would like to just give you a brief update on what we've been doing since we last met. So as a reminder, the work group members are listed here. Some really great neuropsychologists, Rhoda from Boston, Suzanne from Wake Forest, Mary from Mount Sinai, and Sandy from Northwestern. And we also have a number of people who sit in on our meetings listed at the bottom on a regular basis and provide really important feedback. So we thank them as well. So I know we have some new centers here. So I just wanted to give just very brief what our general considerations were for the cognitive battery of the UDS-4. We wanted to make sure we keep the total cognitive testing to 1 hour or less, which we are doing. We wanted to optimize paper and pencil tests to allow time for novel digital computerized measures, and you'll hear a lot more about those options later. And we ideally wanted to plan for at least 2 tests to be given in each domain, potentially pairing a conventional test with a digital measure. So you heard a little bit about the goals and design principles overall for the CTF, but we also had some for the COG working group as well. And I just want to briefly go over those. One was to maintain and enhance the value of longitudinal data that's already been collected since, I don't know, UDS-1 probably. We wanted to minimize participant and center burden. We wanted to have a battery that was sensitive for early detection and longitudinal changes. And we, of course, wanted to ensure that the assessments are comparable across diverse populations. All survey items, measures, and tests should be reviewed for reliability and administration efficiency to ensure the most efficient ascertainment of critical information. And of course, we would make sure that any instrument that we propose will be piloted and validated before ADRC network-wide adoption, including crosswalk studies for harmonization. Like the other working group, our decision-making process includes, discussion amongst our working group, as well as bringing in experts, which you'll hear about later when Andy presents, as well as getting critical feedback from the centers, which we did last meeting in our breakout sessions, and in other ways, people reaching out to us to give us suggestions about different things. So the decision-making process has been fair and robust. So these are the recommendations that we presented last time to continue with the UDS3 battery plus TCOG, VCOG data collection. Last meeting, Gary Chan presented data on the UDS3, showing that it is doing a great job at picking up longitudinal change and measuring cognitive function across the spectrum from normal cognitive function all the way to AD, and also works well across race and ethnicity. So we think continuing with the battery is a good decision. We talked about as a recommendation to add a standardized verbal learning measure, but we're still in discussion about that because we have to figure out how best to achieve consensus, whether we can agree on which tests to use. We did a survey of centers, and there were about 4 or 5 that people are using across the centers. We could possibly adopt a digital measure versus selection of a conventional test. So you'll hear more about that later. We all agreed that we should add digital speech capture. And we have been working on addressing hardware, software, privacy, storage, and analysis solutions. And the digital pen data capture is still on the table. I think you will hear more about that later. So this is the progress that we have made so far. We are going to hear an update on the UDS3 battery plus TCOG, VCOG data collection from Suzanne, representing the Wake Forest team. We are going to have a discussion led by Andy about adding a standardized verbal learning measure and what we've done to think about that, and some other considerations that Andy will discuss. And we have definitely decided to add digital speech capture. The SOP is coming soon. We are working out all the details. We have a great template from Rhoda O that we're going to tweak for our purposes and plan to have it stored at the NACC website. And we're also planning for digital data storage. So that's a really fast, brief update of what we've been doing and what our plans are. Now I will turn it over to Suzanne to talk about remote cognitive pilot update.
Thanks, Lisa. Next slide, please. Yeah, today I just wanted to give a brief update on where we are with the remote assessment pilot and plans for future validation study. So as I think most of you remember, we had initiated a remote cognitive assessment pilot in response to the suspension of in-person visits that was necessitated at the beginning of COVID, but also in response to a need to have such a tool so that we can expand our reach into underserved and rural areas, as well as be responsive to our participants' preferences. So we piloted 2 modalities, a telephone versus a video version, in folks 3 to 6 months before or after their in-person testing. In the bottom left, you can see the characteristics of the pilot sample, which is now complete. We tested about 95 participants. We were able to go across a range of cognitive diagnoses. We did have some diversity within our sample, but certainly not what we would be optimal or we would hope for in our future validation studies. Because we wanted the batteries to be to use identical tests, we restricted them to test with non-visual stimuli, and so we adapted a couple of tests, such as the blind MoCA or trail making, to use the oral trails. And we had 2 versions of the battery, a briefer UDS only battery, and then an expanded battery that included a list learning test. And just to summarize the results for you, we're in the process of writing the paper now, which we hope will come out soon. But to summarize the results, in general, we found that video assessment showed better correlations with in-person than did telephone. And you can see this represented simply in the table on the right, where 8 of the tests listed here had higher correlations for video than telephone modality, whereas only 2 had greater for telephone, and 2 had no real difference between modalities. But in general, we were very encouraged. Most tests had very acceptable correlations, with only a few exceptions, which again, I think we'll need to replicate this in a larger validation study to be confident about that pattern. Next. It was also important for us to assess how participants experienced the remote assessment. And so we asked them to rate the experience on both acceptability, how acceptable it was to be tested this way, and pleasantness, how pleasant it was. And I think, as you can see here, it's a busy table, but in general, participants found the remote assessment as acceptable, as pleasant, or easier and more pleasant than in-person testing. And I don't know what that says, but we are very encouraged that participants seem to respond positively to the remote assessment. And in fact, when we asked them, how likely are you to choose phone or video assessment over in-person assessment, with a 10 being very likely, I think you can see in the bottom right panel, there was a pronounced preference for the remote assessment. Now, again, it's hard to know how much this is temporally linked to the current COVID situation, and we'll be testing that. But I suspect not. I suspect that there are going to be a significant number of participants who actually prefer the remote cognitive assessment. Next, please. And so for my last slide, I just want to remind folks, I think we mentioned this in the spring meeting, but remind us again about the cognitive assessment validation R01 that is being led by co-PIs Bonnie Sachs and Steve Rapp at Wake Forest, which aims to develop a psychometrically sound tool for remote assessment, and will compare cognitive scores and adjudicated cognitive status for a video assessment versus in-person for 500 ADRC participants, with a big emphasis on a diverse group of participants, both from racial and ethnic perspective, as well as from a cognitive diagnosis perspective. And they'll be tested twice at a baseline and a 2-year follow-up, so that we can look at longitudinal change. We've had 12 ADRCs that will participate, that are listed here for you. And we haven't heard definitively. We did receive a very good score. The application scored at the 7th percentile. So we're optimistic, but we have not received our NOGA yet. If we are successful, we project to start in the spring of 2022, and we're beginning to think about potential ancillary studies that might be potentially attached to the architecture of the validation study. So I'll stop there, and thanks.
Great, thanks very much, Suzanne, and thanks so much to the Wake Forest team for all of the Herculean efforts to bootstrap that remote assessment. In the next, I think we have a couple of minutes left, just 2 slides, although they're busy ones. I want to tell you about what the Cognitive Working Group has been doing in conjunction also with Rhoda O's and the technology work group to look at digital assessment. And then there'll be a session on this coming right up, so I don't want to overlap with that. But essentially, the committee has been looking at the various options that are out there. And everyone knows this is a rapidly growing area, digital health, and within that, clinical and cognitive assessments. So we've been looking at platforms, at tablet-based measures, at mobile measures, and other strategies, trying to get a handle around what are the options and what are the potential, what's the timing and potential benefits and limitations of some of the different platforms and approaches. So we've been having meetings every other week, and really appreciate all of the efforts of the whole committee and our, the NIA folks and NACC folks who have been attending all of these, and the guests who have come in and presented to us, some of whom you'll hear from today. So, we've had presentations from Kate Posen, who you're going to hear from shortly, and Adam Stefaroni as well around the TabCat, the Richard Gershon from the NIH toolbox, and a little bit about the development of the mobile toolbox. Martin Slawinski has met with us and talked with us about the mobile test battery that he's developed and is developing further. We recently had sessions with Huma, where they have a digital speech capture and word list learning paradigm, and Novoik most recently as well, where there's a digital story recall with multiple alternate forms and speech capture and analysis. Then there are many other approaches out there. We welcome input from the community, as we've mentioned before, about strategies and technologies that are available, but we really appreciate all the input that we've gotten so far. So probably all of this is moving toward the direction of a pilot study to demonstrate the feasibility, probably in several centers, and then ultimately the goal would be to have something that would be made available to all of the centers on a network-wide basis, but that really will take some significant pilot work following the principles that Lisa outlined earlier. So we're trying to ensure that we welcome input from the community. This gives you a snapshot of what we've been looking at so far. We're also talking with ADNI, that's going through a parallel process of looking at a lot of different technologies that may be especially relevant for clinical trials and longitudinal follow-up. Next slide, which is the last one. So what are the other things that we're doing? Well, digital assessment we spoke about. You'll hear more about from Rhoda and the group of presenters. We've been thinking about the goals, whether the goals should be the same as for conventional testing, or there's really different opportunities and different challenges with digital, which I think you'll hear about shortly. And we're trying to get more clarity on that. Some of the other things Lisa mentioned, the issue of a standardized verbal learning test or word list test. There'd be some real advantages to that. And I guess the rubric is really standardization to one instrument versus post hoc harmonization. And there's a consortium now focusing on cognitive imaging and other harmonization, but if you've standardized to begin with, there's a lot less work to do that. And then, we've been looking at through Mary Sano, especially right now, looking at the issue of story or narrative recall versus word lists, and what's the relative incremental information yield from doing two versus doing one alone. That needs to be looked at in more detail. There's a topic that I won't say more about now, but it's come up and been on the table, is should the ADRC network develop its own screening tool that might be MoCA-like, or perhaps some other more innovative screening measure for early detection. The issues of sensory function that came up during the clinical measures, which have been ported over to our working group in terms of vision and hearing, which are so relevant to both remote and even in-office testing, and maybe identifying whether there's a need for a specialized battery to pursue sensory changes. Simple issued one bullet point here: neuropsychiatric measures. As a community, are we happy with the performance of the the NPI and GDS? The way that we're capturing that information, or should we be doing something else? And the relative costs and benefits of doing that discussion is really just beginning. And then, as Lisa mentioned, the issue of early cognitive concerns or subjective cognitive decline. There's a lot of research in this area. It would be a whole session to go into any details. There's an international SCD initiative and a Alzheimer's Association PIA addressing this. There is a consensus statement that's probably one of the more recent captures of this that's by Frank Jessen et al. In Lancet Neurology. I'll call your attention to. So the final point is that we're really beginning to address this and looking for whether there's opportunities for some brief standardization of approach to this. And I'm running over and we'll stop here, but thank you all. Next up is the technology session, and Rhoda will, I'm sure, introduce the speakers.
Thanks. Thanks a lot, Andy. Unlike the other UDS work groups, there's actually no prior tech UDS 3.0 that we're going to build from. So what we're going to do in the first couple of presentations is maybe preview what elements of UDS 4.0 is going to look like. And then the next two presentations will actually feature ways in which digital data can be analyzed when it's put in the hands of those with data science and AI expertise. So to get us started, we're going to start with Kate Posing, who's an associate professor at the UCSF Memory and Aging Center and a faculty member at the Global Brain Health Institute. And she's really been doing tremendous work in the development and validation of a whole suite of tablet-based cognitive assessments, a subset of which will be part of UDS 4.0. So Kate, all yours.
Thank you, Rhoda. It's great to be here and to share our work with TabCat and the potential value of some of our tests for inclusion. The tablet is an ideal interface for cognitive testing. It offers all of the advantages of computerized testing, including standardized administration, dynamic stimuli, precise response capture, and automated scoring with no need for data entry, which our coordinators love. And at the same time, this tablet interface is comfortable with its large screen size and easy response format, even for older adults who've never used computers or smartphones. Tests can be examiner administered or proctored self-administered, and we have tests in both of those formats. So we developed this software platform and the tests on it, TabCat, out of response to noticing that there was a growing number of commercial products that didn't have the scientific backing that we thought that they should have. We felt it was important for ADRC neuropsychologists to lead the way in the development and validation of cognitive tests in this format. So a set of the tests on TabCat are being considered for possible inclusion in a UDS 4.0 pilot. And I'm going to tell you about some of those today. So these are the tests that I think we should think about for possible inclusion. I don't think we would include all of them, but I wanted to give you a bird's eye view of the tests we're looking at. We have two tests of executive functions and speed. One is match, which is similar to digit symbol. Another is a flanker. We have two different associative memory tests. We have a couple socio-emotional tasks that are sensitive to FTD. We have a visual spatial task, line orientation, that's adaptive. So it gets harder when you do well and easier when you do poorly. So we quickly and precisely estimate spatial threshold. And we have a rapid naming test. Participants view a series of easy to name objects and we see how many they can name in just 1 minute. It's much more sensitive than the standard confrontation naming paradigms. All right, so now I'm going to dive into the science for some of these tests, focusing mostly on the brain health assessment. These tests are match, the one that's like digit symbol, one of our associative memory tests called favorites, and the line orientation. Sometimes we also think about animal fluency as being part of the brain health assessment. So we have a language generation test there too. You can see we're sampling the key domains that are important for early detection and monitoring. So as we reflect on Andy's comment that the ADRCs might develop a screening tool like the MOCA, I would argue that this might be a good option for us. So showing you now some of the science we've done on these tests. These are voxel-based morphometries, and we look to see if the brain health assessment subtests have expected relationships. We found that MATCH, our executive function test, had relationships with dorsolateral prefrontal, parietal, basal ganglia, just as you would expect for an executive function test. Our line orientation task with right parietal, and our associative memory task with the bilateral medial temporal lobes, including the full extent of the hippocampi. Early detection is really one of our central goals. And here we compare the detection of the brain health assessment compared to the MoCA. So you can see in dementia with the blue line, we show almost perfect separation of dementia from controls with the brain health assessment. You can see the MoCA in orange. The difference becomes even more apparent when you look at the MCI sample. So much more sensitive. And keep in mind, this is about the same testing time, 10 minutes. We've had the opportunity to validate the brain health assessment in some diverse populations and have published on 2 different Spanish-speaking samples. Here's a Cuban sample where we show almost identical results comparing the MoCA and the brain health assessment for classifying dementia versus controls here, and then MCI versus controls on the right. The brain health assessment is reliable over time. So we see that it's very stable over time in controls in yellow. We can see MCIs, about half of them decline over a year, about half of them remain stable, which is probably what you would expect, whereas dementia patients, the majority of them do decline. So much more precise than a paper-based assessment like the MoCA for detecting change over time. And this is a sample of just cognitively normal individuals. These are people who we have no concerns based on all of our other assessments that they have anything wrong with their brain in terms of neuropsych testing, interviews with informant, et cetera. But the controls who are amyloid positive show decline on the brain health assessment over time in comparison to the amyloid negative controls. This is not something that we can pick up on with MoCA. And here we're looking just at patients now and dividing our MCI patients and our dementia patients by their amyloid status on amyloid imaging. And we find that the match and favorites test, those 2 tests that are so critical for our brain health assessment, the memory test and the executive test, they really pick up on something important for Alzheimer's disease. And in fact, their association with A beta positivity is evident even after you control for disease severity. So, and having tests like this is really critical as we move into this new era of treatment. If we focus in now on those patients who are amyloid positive, we look at the relationship to regional tau deposition and find that the memory task favorites is associated with hippocampal tau deposition. Match with dorsolateral prefrontal tau deposition, and the line orientation with dorsal stream occipital parietal deposition. We think a lot about how to use our tests to improve care for patients with the Detect CID consortium in particular. And we have integrated the results from the brain health assessments into EPIC. So in our primary care clinics now at UCSF, after the medical assistant administers the brain health assessment following a primary care visit, the results are instantly available in the form of a note that looks like this that guides the primary care provider on next steps in care. And by implementing this in our primary care, we've been able to increase their diagnoses of MCI and dementia by 50%. I can't give you a TabCat presentation without highlighting some of our really beautiful social tasks. Although I know that's not the main focus of the tests we're selecting today, this work is led by Kate Rankin. In particular, the DART test is a test of emotion reading. The participant watches 12 short videos and has to identify the emotion that's being conveyed based on the body language and the tone of voice. Performance on this test is associated with right temporal pole and orbital frontal anatomy and connectivity. And our social interaction vocabulary test, which is about understanding social semantics, is associated with medial orbital frontal anatomy. So in closing, TabCat is really developed by a team of neuropsychologists, all part of the ADRCs. We've been working on TabCat since 2014, and since that time, Kate Rankin and Joel Kramer and myself have met every week to work on it without fail. It's been a wonderful collaboration, and Elena is now an integral member of that team. Elena Soy, you see, she was the first author in some of those key publications. Adam Staffaroni helps us think about how to make sensitive and reliable composites of executive function measures. And Sarah Farias has helped us think about how to measure everyday cognition as part of our measures, and helped us collect data on TabCat at the Davis ADRC. And the newest member of our team is Leslie Gaynor, who comes to us from the Florida ADRC. So thank you for the opportunity to share some of our work today. Rhoda?
Thanks, Kate. That was great. So next up is David Bates, who is CEO of Linus Health, and in some former life was also a PhD in chemicals and materials engineering. And he's going to present to us a test agnostic platform that we're going to be piloting for the UDS 4.0. I think most importantly also is that this platform is customizable so that each center could make it fit into their needs. So David.
Thanks, Rhoda, and thank you for everyone for having me here to present Linus Health. And so I'll just flip through some slides. So what Linus Health is, the platform, Linus, as Rhoda mentioned, it's a unified digital platform that's used to collect, aggregate, and analyze clinical study data. All the assessments currently on the platform are validated, both in peer-reviewed journals and several of them with FDA registration and CE marks. And so they're available right off the shelf and can be used in clinic or in studies. One thing I really appreciate about the Linus platform is the multimodal approach. So we're taking standard neuropsychological assessments, but also using the digital versions of them to assess not only the test results, but the whole test taking process, even pulling in some context, so that it can be reviewed time and time again. So not only the results are reviewed, but actually how they went about carrying out the assessment. And I'll talk more about that in a moment. Linus was designed to be convenient, low-cost hybrid collection of data, so you can run it both in clinic and remotely, and it all ties together seamlessly. And it's designed from the outset to be easily integrated into existing systems or devices. And so it can integrate into REDCap so that it pulls data and feeds data as you need it, as well as integrating other sensors or devices or whatever you need to really carry out your study. So I think the major proposition to the ADRC around the ADRCs around the country is that it really streamlines study preparation, as well as unifying all the different things going on in that study during the assessment. So you can pull in, medication management, EHR records, or existing study records. It's got an API framework, so that's why we can work with TabCat, as Kate just presented, or CART. We can also pull in existing apps through deep linking on the App Store. You can digitize question responses or clinical outcomes assessments. And then as assessments go, we really went to great lengths to pull together, as I said, multimodal. So really analyzing all kinds of drawing behavior, even eye tracking, even we have spirometry in there, health records analysis, gait and balance, dual tasking, reaction time testing, voice and speech. So these things are currently available right now. You could just select them and integrate them into your study. And I should mention on voice and speech, I saw Nevoic on a slide earlier in the presentations. And we currently are integrated with Oral Analytics, Winterlight, and Nevoic. And we plan to bring in more. So there's a vast array of speech assessments that can be chosen from. And again, all of this is managed on a single platform. So all the participant sees, or the administrator, is a calendar or a list of participant numbers and who's supposed to be taking the test, and it just pulls up their assessments that they have to run that day or in that period of time, and it's all seamless with a good interface. So I think another one of the major benefits of the Linus platform is that it's incredibly secure. It's asymmetrically encrypted on the device, so that even if the device is lost or stolen, either the one in clinic or remotely, no one can access that data. So it's not decrypted until it hits the cloud, where it's decrypted and analyzed. Another key point is that these assessments are immediately available. So they run right through. As soon as they hit a gateway, they're analyzed in the cloud, and then the results are right in the dashboard. And so you can look at them right away, or you can keep reviewing them. And these, as I said, these analytics are performed in real time. And so just as an example of some of the capabilities, you know, the DCT clock is on the platform. And it was shown earlier this year, it was reported that it's incredibly sensitive, especially in the early detection of amyloid burden. It's associated very well, even better than the PAC and the MMSE for those who are clinically normal. But it's not only a very sensitive tool, it's you can replay it and look at, you know, how did the person do? And so if it was administered by a research assistant or whatever, the PI can look at it and say, why did they score this way? Because, you know, they, according to classic CDT criteria, this is a 3 out of 3, but they scored very well. And indeed, this person had very early Alzheimer's disease. And so you can review the assessment, not only for the sensitivity, but on the dashboard, you can review for specificity. So we just saw this person had early Alzheimer's. This person had Parkinson's with diffuse Lewy body disease. And by being able to review it right on the dashboard, you can see the little tremors and you can see the reasons why it was scored the way it was. And of course, all the data we present to the ADRCs, they get the raw data, so you can each analyze it with your own methods or combine it with other assessments you're taking. But it just gives you all that power at your fingertips. And again, with the customization that you desire, communications with remote study participants, it's all being built into the platform from the outset so that you have that flexibility while conducting your studies. Again, voice data, I saw that voice is a major initiative in this next series, the UDS. Not only do you have the ability to do transcription and time-stamped voice, you can just listen to it again right away. How is it being collected, or is there good quality? So all of this is available again at your fingertips, and you can download it and do your own analysis on the raw data. So one potential ADRC hybrid protocol is to unify these batteries with, you know, digital question and responses. I'm sure everyone has these these questionnaires. They can be digitized to try to collapse documents management and workflows. So they can be digitized. You can run a series of assessments, standard assessments, but getting all that temporal data as well, not only the results. As well as, you know, as Kate was sharing on TabCat, bring TabCat in, make it part of the standard in clinic, as well as the remote. On the remote, we can do a lot of digital voice, gait and balance. TabCat does CART, and again, we can push questionnaires. You can do standard, the Dana battery. So it's really just, I guess the takeaway here is that not only are these validated and incredibly customized, it really, you can capture the process, and you can really customize, not only do the standards across all ADRCs, but you can customize it to your own hypotheses and what you're intending to find or looking for in your particular population. A little bit about the applications themselves. In order to go hybrid and remote, we do have the in-clinic test administrator app that research assistants or medical assistants would be using to collect data from participants in clinic. There's the at-home participant app, which someone just gets a notification. Your assessments are, you're supposed to take some assessments today or this week, which can be customized. And then, of course, we have a link to a study partner app for those who are suffering a little bit more, they're a little bit more impaired. You can engage a study partner to help remember to carry out the assessment remotely and communicate with them to get that hand, to get some help remotely. And then I think another key thing about the Linus platform is our commitment to customer success, which is not to just provide a lot of technology at your fingertips, but really to support you every step of the way, both in your planning, in providing the equipment if you don't have it yourself, and then training up research assistants, and then really coordinating to go live and provide that ongoing support and making sure you're getting the most out of the study that you've put so much into design and carry out. And with that, I think I'm at time.
Great, thanks, David. So next up, and I actually have to do the share screen. So let's see if I can do both. Let's see. So next up is Vijaya Kolachalama, and he's assistant professor in the section of computational biomedicine in the Department of Medicine at Boston University. And he's also part of the Department of Computer Science and Computing and Data Sciences. And what's been interesting for me in working with Vijay is he's actually really made me completely revise how I think about how digital data can actually be analyzed. So let me tee up Vijay's slides here, and you're up now.
Thank you, Rhoda. Thank you for giving me this opportunity to talk about this topic, which is kind of related to how latest advances in machine learning and AI can really allow us to push forward a new paradigm in data analysis. The main point that I'm trying to highlight on the title is that we as a community, we need to start thinking about how novel approaches to data analysis using AI by first redefining the way we think about data itself. Because I think some of us are already doing it, but I think more needs to be done. We are a lab of computer scientists working on building novel AI approaches to address various aspects related to AD assessment. We primarily focus on methods development by viewing data as sort of this unprocessed entity. Next slide, please. By unprocessed, what I mean is obviously raw data. And by raw data, what I mean is the data that is native and that I can directly get from the scanners before an expert. Move forward, please. Or a software that even begins to touch the data before even an expert or a software can begin to touch the data. We believe this is exciting because data in its native format has the most information. For example, the DICOM files in images, the Nifty files with regards to the latest digital devices, such as speech and next please, like Apple Watches. Data just simply can be raw voice files in just simply the native format, which is the WAV formats. I must note that the information in these raw files might be very, very unstructured, but the value of mining such data can be incredible. So our lab is primarily interested in taking this raw data, or this crude oil, if you will, where we actually start and then start our analysis. We have built an infrastructure around it. We continue to tackle problems at various levels because we feel like we have the most flexibility with this setting, the setting of actually starting from the point of getting that raw data. Next slide, please. So one of the first things we did was we collected data in these native formats. Obviously, most of you here know these cohorts. The one that we are most proud of is the Framingham Heart Study, because we use the headquarters for this cohort, and we have gone great lengths to collect, to process, to harmonize the data in this native formats. The best part of this data collection is that we were able to test our automated approaches and these computational workflows on all these cohorts. This kind of exercise gave us huge confidence that we can work on both retrospective as well as prospective studies. Our computer programs are robust and generalizable, and we are excited to sort of test them on future studies as well. Next slide, please. So now that we have the raw data from all these cohorts, the big question is what we can do with this data. Right? Like I noted in the previous slide, we had to spend a significant amount of time to sort of really build computer scripts that can process the raw data. Once we did that, we focused on building analytic tools that can directly learn from these data to predict various outcomes. Here are two examples. Move forward, please. The one on the left is the first proof of principle that raw voice recordings collected from neuropsych testing sessions done at Framingham Heart Study can be used to build a deep learning approach that can assess the dementia status on those individuals. And one of them is the participant, and one of them is the person who's actually asking those questions. The one on the right is a fully automated approach that can directly process volumetric DICOM files of the brain images to predict Alzheimer's status in individuals. This was a paper that was published last year in Brain. The one on the left was accepted as part of a special issue focused on AI and dementia. So these two examples, essentially, what they show here is that we have a good handle on raw data. You could build hands-free approaches to assess interesting things from the raw data directly. The key point is that we probably don't need to ask the experts to really spend time to manually curate information. So I think this is really huge because if we want computer models to be scaled around the globe, we can't expect the experts to sit down manually and work on all these cases. So things need to be automated, and we need to think about automation from the standpoint of sort of really starting from that raw data and then doing things from that, and then establish workflows that can be generalizable. And that starting point for that automation is obviously when we get the data, which is not touched by anyone. Next slide, please. So in order for us to believe what we are doing and how we are doing and everything is right, we did not simply want to build the models. We also wanted to spend some time to make sure they are sort of validated. Here is an example that shows that predictions from a deep learning based assessment of volumetric brain scans, they closely match the neuropathological findings of Alzheimer's disease pathology. Such confirmations actually allowed us to make sure that our models are grounded. And in summary, I think what I wanted to say here on this slide is that validation of the models is very important. And we spend a lot of time to focus on not just model development, but also validating those models. Next slide, please. So finally, and very quickly, I want to talk about data harmonization. This is, again, a very important topic, especially if you're dealing with different forms of data, multimodal, if you will. This is a huge topic because there have been several papers that have been published and progress has been made in general. The simple point I want to make here is that when you have access to raw data, then it is possible to learn from that raw data directly to not only just predict outcomes, but also to learn from the data itself to see how, for example, in this case, how scanner-related differences or, let's say, magnetic field strength-related differences, or more broadly, instrument-related differences can be normalized, even before we do data analysis. So I think the point here is that when we have the raw data, we can probably think about approaches to data harmonization, and then even also think about data analysis. So this was, again, another recent paper that was accepted in the same special issue focused on AI and dementia. Next slide, please. So yeah, again, in summary, the key points are that data in the native format is what we like, and I think that needs to be harnessed as it has the most information. Recent advances in machine learning, especially deep learning, has provided us with these strong analytic frameworks to directly tackle raw data. And we spend a lot of time to build these automated frameworks because I think they are the ones which I think personally have the biggest potential to be generalizable. And AI can also be used effectively to address issues related to harmonizing different forms of data, whether it's images, voice, you name it. And I think the focus needs to also be on not only just building those methods, but also making sure that these methods are comprehensively validated. And final slide, please. So I just want to quickly thank my lab members. I think the students who have been the ones who sort of really work very hard to build all these approaches, they're all computer scientists. And I think the visionary who actually made this all possible is Dr. Rhoda Oh, who gave us the chance to work on these exciting projects. Thank you.
Great. Thanks, Vijay. So next up is Yanis Pashlidis, who's a professor of electrical, computer, and biomedical engineering and director of the Center for Information and Systems Engineering at Boston University. And what I have to say is what I really love in working with Yanis is I pose these problems to him in terms of all these limitations I have about what I can do with digital data. And then he goes away and comes back a few months later, and he has a totally mind-blowing solution. And so I'm hoping that he will do the same for you today. So Yanis.
Thank you, Rhoda. Thank you for the introduction, and it's a great pleasure to be joining today. So I'll talk to you about algorithms for dementia detection based on digital biomarkers. And I'm guessing I'm not going to spend a lot of time on this slide. I think people that have joined in this session understand that the current practice of in-person neuropsych exams is a practice that is not scalable. And another aspect of it is that it accentuates disparities in healthcare, as it is clearly available only in settings that have ample resources. And one would be interested in more cost-effective diagnosis procedures that can be, and that I think is an important aspect, conducted remotely, that are globally available and are independent of language or local social norms. And in this talk, I'll talk to you about two specific digital data sources that we have exploited in our recent work. One is image-based and the second is voice-based. So starting with the image-based, we took the venerable clock drawing test, and we used subjects from the Framingham Heart Study. As input, we have the command clock. So this is the clock that is drawn where the subject is just asked to draw a clock, an analog clock, and then the copy clock is the version where the subject is actually shown a clock and is asked to copy the picture that is being shown. And then we process this through a so-called convolutional neural network. So this is a deep learning model that has actually been trained on many images to be able to recognize various objects in these images. And out of this, we get a score, which you can think of as a likelihood for the patient having dementia. So we retrain using a transfer learning approach, the CNN. And then in addition to the score that is produced by this deep learning processing of the images, we train an ensemble model, which you can see here. And this ensemble model has in addition some demographics, so the age of the patient and also the educational level. And the task at hand here is to predict dementia, so whether a patient is normal or has mild cognitive impairment or dementia, and we have lumped together these two classes, MCI and dementia. So this is a work that we just published in the Journal of Alzheimer's Disease. Let me also mention that in the images that we use, we introduce various distortions to these images. We wanted to, in some sense, simulate a setup where an image is being drawn on a piece of paper and is captured by a smartphone and is being submitted, rather than an image that is being drawn in an in-person setup. So even though we don't have such images that are captured by a smartphone, by introducing these types of distortions, we hope that this gives you a sense of what could be done with images that can be captured in that specific way, in that specific modality. So I'll show you results. So the ensemble model has an AUC of very close to 92%. And if you restrict to subjects that are older, greater or equal than 60 years old, then you get a slightly lower AUC of 86.6%. If you just use age, as the only feature, you get actually pretty good AUC at 89.3. But let me just point out that the difference becomes significantly greater if you focus on the parts of the ROC curve that you would like to operate. So for instance, if you set your false positive rate threshold to 10%, then the ensemble model gives you a true positive rate of 73.1%, where just the age model gives you a much lower true positive rate of 59.5%. And obviously this has implications on, for instance, if you are looking to recruit members for a potential clinical trial, you will miss many more using just the age model versus using the ensemble model that takes into account the images. And in this figure on the right, you can see the relative importance of the various features that enter in the ensemble model. So the age is an important feature, and also the command clock appears to be more informative than the copy clock. And then, as probably expected, the higher the educational level, then the lower the likelihood of the person having MCI or dementia. So in the next study, this is just a very recent, we've just completed that and we submitted to publication. We worked with voice recordings. So in the Framingham Heart Study, they record digitally the entire neuropsych exam. We had about 1,000 or so subjects and their recordings of their neuropsych exam. And from those, only a small number, 133, the recordings were manually transcribed. And what we were interested in doing is to develop a completely automated pipeline that takes as input the recorded interview, and then as an output, you produce an assessment of the dementia status. So the approach that we used is the following. So we automatically transcribed the interview using speech recognition. So specifically, we used Google Speech. And out of that, a transcript gets produced. And then natural language processing takes over. So we take the transcript and then we diarize it automatically using natural language processing. So we separate the parts that the interviewer speaks and the parts where the interviewee speaks. And then we also do sectioning of the interview. As you probably know, these neuropsych interviews have different sections depending on the type of test that the subject undergoes. So in the Framingham case, there are 8 different tests. And we label the parts of the text that correspond to different parts of the different tests of the interview. And then all of these, the sentences that correspond to each one of the tests get encoded into a long numerical vector, and that vector now becomes input to an ensemble model that together with some additional demographic information produces the final prediction. Let me just mention that in my group, we have done quite a bit of work on natural language processing, so this was sort of a natural path for us to take. And specifically in some of the earlier work, we have found ways of infusing biomedical knowledge into more general, generic language models. So let me show you results. So we looked at 3 different tasks. So in the first task, we want to differentiate subjects that are normal versus ones that have dementia. So we call that dementia detection. In task 2, we want to differentiate normal or MCI versus dementia. You can call that non-dementia detection. And in task 3, which is arguably much harder, we want to differentiate between normal versus MCI, and we have excluded those who have dementia. So you can see that the approach that I described, so using the encoding of the different sections of the interview, plus demographic information, sex and age of the subject, gives you a fairly high AUC of 91.2% for task 1, 87.1% for task 2. We can do a little bit better if you add, so this random sampling here refers to just randomly select some sentences from the entire interview and also process them through natural language processing and throw them in the mix. So you get a little better AUC. And then for the third task, which is much harder, normal versus MCI, if you add ApoE information, you improve a little bit the performance and you get an AUC of about 74%. Let me just say that it's actually possible to assess the relative importance of different sections of the interview, because in the ensemble model, we can compute weights for different sections of the interview. And there are some interesting conclusions that one can derive. Some tests are more useful in differentiating between normal and dementia, and some other tests are more useful in differentiating between normal and MCI. And I will stop here, just thanking people in my lab, and of course, collaborators, Rhoda, who has played a really important role in this, and many others, Vijaya, that we have collaborated in this work. Thank you.
Great, thank you very much. That's great. So I'm actually, I know we're kind of running late. I'm sorry about that, but I do have a few slides that I wanted to share. And so I think in these last 4 presentations, you've got a chance to see what's on tap and the type of things that are actually really possible now. And what I'd like to do actually is get us to really stretch beyond our mental comfort zones. Oops, sorry about that. Let me, a little bit too soon there. And really think about embracing the shifting of our current mindset. And I think if we can do this, we're actually going to shift our research paradigms. So to start this mind shifting process, I like to remind everyone there was a time where we thought the world was flat. And then as we moved to discover what we came to understand as our round Earth, we actually started to look beyond. Beyond our boundaries and set our sights on the moon. And even though we had absolutely no idea how to do it, we set the goal of landing there. And we found a way to make our way over there to a great discovery. Now, the satellite has taught us that we still actually have a lot left to learn about our own Earth. And even as we continue to explore that, we're now targeting Mars. So now bringing this back to the digital area, to today in the digital era, I want us to think about our goal is trying to move from the world we work within and think about how to get to someplace that's much more difficult. Now, often in science, we're not unlike ants on the hunt, where we will follow a path laid down by our peers, we'll follow it faithfully, and when we do that, we actually can miss opportunities that are off to the side. So I want to actually introduce you to the Argentine ant, which is a warrior ant that over time has built into a super colony that stretches actually across 5 continents. Now, what's interesting about the Argentine ant is if one ant from one continent and another ant from another continent and yet another ant from another continent, if they all meet up together, they actually will recognize each other, and they will recognize that they're all coming from the same super colony, and they will start to work together. And then as they complete, their initial tasks, they will go forward in new directions, and they'll do it together. So these ants don't see borders and they don't see divisions. They understand that they share the same purpose, and they will quickly blend together to find their next landing spot. So that's what I think we need to figure out how to do. We need to split off from where we are, come together in this new digital era, and chart a new course. And the new course I actually want to highlight for the remainder of this talk is the realm of digital biomarkers. Now, when I first started using those terms together, digital and biomarkers, I was actually admonished and told I couldn't use those 2 words together, that they didn't fit the traditional meaning of what a true biomarker is. So now it's interesting, 7, 8 years later, the term digital biomarkers actually become all the rage. And I would say in its most common use, its full potential hasn't been yet to be realized. So the question is, what should a digital biomarker really look like? Well, for starters, it's most likely to be comprised of multi-sensor inputs. There's not going to be a single device, and there's not going to be a single application that's going to be sufficient. And given the complexity of the multi-sensor input, unquestionably, data science and AI is going to play a critical and central role. Now, importantly, what will come out is not going to be a single metric or a single pattern. Instead, what will emerge will be a flow of dynamic, unique combinations of sensor patterns where no one pattern will be a reliable indicator, but in the aggregate is going to provide an accurate assessment. Now, I kind of want to remind us that we already do this clinically. Whenever we do a patient intake, we ask a person when they first started to notice symptoms. And the person might say they forgot to turn off the stove, they got lost going to a familiar place, they couldn't remember where they stored an important document. It's going to be a series of different behaviors that combine together to nonetheless reflect a memory impairment. But as we all know, here's the thing. The person doesn't always forget to turn off the stove, doesn't always get lost, doesn't always misremember where they put something. And if you ask family members the exact same question, they're probably going to give you different behavioral examples, like a missed bill payment, unusual repetition of stories, forgetting about a key family gathering, et cetera. So that's what I think digital markers are going to have to mimic. These scattered and sometimes seemingly random patterns will nonetheless come together and reflect accurately and reliably a prognostic or diagnostic indicator. Now, in defining what should be a digital biomarker, we're going to need to work closely with our regulatory agencies to get the approval necessary to move it out of the research context. And critically, we're going to need to not work with current regulatory pathway protocols, but rather work closely with these agencies to figure out what are the new methods of validation. And to get to this digital biomarker reality, we're going to have to shift the directionality of our thinking. So currently, the scientific method is driven by a left to right approach, which involves having a hypothesis, testing it with well-developed and well-defined methods. And I will tell you, I don't think this approach is going to get us to the digital biomarkers that I just described. So about 13, 14 years ago, I decided to start with a problem and then work backwards to figure out how to address it much more effectively. So going forward, I think we need to change directions. We need to do things less linearly and less tied to what was, and focus instead on what might actually work and do that. And if we do that, I think we'll move toward the reality of the true potential of digital markers. So I'm going to end this talk in this session about the future by looking back to the past. So in 1948, no one knew why people were dying of heart disease and stroke. And the concepts of high blood pressure, high cholesterol, high blood sugar, they didn't even exist back then. The concept of risk factors itself didn't even exist. So the NIH took a bold move of commissioning a 20-year study with no existing roadmap, with none of the knowledge and analytic tools we take for granted today. And from that, that's where the Framingham Heart Study emerged and really thrived, essentially creating the field of preventive medicine. I think aided by our technological tools available today and the future, all of us who comprise this ADRC network are poised to do for the brain what Framingham did for the heart. So I want to thank you for your time and your attention. I want to give a special shout out to my colleagues, Spencer Lowe, Zach Pock, and Philip Jung, who came together like Argentine ants from different corners to help me put together this presentation. Thanks a lot.
All right. Well, we are a couple of minutes over time, but I think that's a pretty grand success for all of the great amount of information that we've spilled into the last 3 hours. So thank you everyone on the clinical task force for your brilliance and sharing so coherently today. And we look forward to learning more as all of you continue to meet and progress.